Welcome to the deep dive. Today we're on a pretty critical mission, breaking down the blueprint for personalized medicine and cancer treatment. We're not just, looking at the tools. We are really getting into the weeds of the scientific and regulatory process behind developing these molecular diagnostics that are completely changing the game.
It's arguably the single biggest leap forward in modern oncology. We're finally moving away from what was, well, frankly, an era of educated guessing.
The empirical Freeman.
Exactly. You were looking at average response rates of maybe 25% for general chemo. The whole goal now is what we call rational pharmacotherapy.
Okay, so what does that mean in practice?
It means getting the right drug to the right patient at the right dosage and at the right time. And molecular diagnostics are the engine that makes all of that specificity possible.
So for this deep dive, our source material really lays out the essential knowledge for any medical professional getting into this field. We're going to distill it down into three big areas for you. First, the molecular tools themselves, so IHC and ISH. Then that really complex dance of co-developing the drug and the diagnostic. And finally, some key clinical applications in cancer and pharmacogenomics.
And to really set the stage, you have to look back at the historical pillars that got us here.
Right, this didn't just appear out of nowhere.
Not at all. I mean, the first true success story goes way back to the 1970s with the estrogen receptor.
ERPR.
That's the one. The logic was simple. If a tumor had those receptors, we knew that tamoxifen, an anti-estrogen therapy, had a pathway to actually work.
So that was the first step. That was stratification. But the real paradigm shift, the one that basically forced this whole parallel co-development model on everyone, came with trastuzumab.
Absolutely. Trastuzumab, which most people know as Herceptin, it only works in about 20 to 25% of breast cancers.
The ones that overexpress the ATR2 protein.
Or amplify the gene, yes.
Yeah.
And that was the moment the FDA sort of realized, wait a minute, the drug is completely useless if you can't find that 20% of the population.
Which meant you needed the test.
You had to have the test. It required the co-approval of a companion diagnostic, the HercepTest. And that moment forever locked the fate of the drug and the test together.
Okay, so let's unpack the toolkit. When we talk about a biomarker, there's a kind of classification system you need to get your head around. How do we break down their different functions?
We generally put them into three buckets. The first is diagnostic. That's pretty straightforward. It's used for disease classification, maybe staging.
Simple enough.
The second is prognostic. This one gives us information on the likely course of the disease, how it might progress if we don't intervene with a specific drug.
And then there's the third one, which feels like the highest stakes category from every angle, both clinically and for regulators. The predictive biomarker.
That's the one. What makes it so unique is the direct, strict link to a specific treatment.
So it's not just about the disease course.
No. It identifies patients who are statistically likely to benefit from or, and this is critical, be harmed by one specific drug. The success of that drug literally hinges on the test's accuracy.
Which means the regulatory risk is through the roof.
It's the highest risk category. A prognostic marker like ER-alpha might be, say, an IVD class 2, moderate risk.
Yeah.
But a predictive companion diagnostic, always under the absolute highest scrutiny.
Let's get into the tech then. Starting with immunohistochemistry, IHC. It's targeting proteins. On the surface, it seems simple. You're stating a slide, but the analytical rigor behind it is immense.
It really is. When you're developing an IHC assay, you have to prove two kinds of specificity. The first is sequence specificity.
Okay, what's that?
That's just the antibody recognizing the correct sequence of amino acids, the right protein.
Yeah.
But the second one, modification specificity, is much trickier.
Modification specificity.
Yeah, this is about detecting a change that signals the protein is functionally active. Think of it like phosphorylation or acetylation. It's not just about seeing if the protein is there, it's about checking if its light switch has been flipped to on.
So how on earth do you confirm that? How do you prove the antibody is only seeing the on switch?
You have to use some clever biochemistry. For example, you might treat the sample with a phosphatase. That's an enzyme that removes phosphate groups.
And if the stain disappears?
Then you know your antibody was, in fact, recognizing that phosphorylated active state. You can do similar things with drug inhibition models. The whole field is now pushing to get to single molecule detection to make IHC truly quantitative and move past just a semi-quantitative score.
Okay, so from proteins, let's move to the genome. We use in situ hybridization, or ISH, to look at the underlying DNA and RNA.
Right. ISH is how we spot chromosomal abnormalities, amplifications, deletions, all those things right inside the cell's nucleus in the tissue. The workhorse here for a long time has been FESH.
Fluorescence ISH.
Exactly. FESH is fantastic because it allows for a multicolor probe, so you can look at, say, HER2 status, but at the same time you can look at another gene like TOP2A and a control on chromosome 17. It gives you all this context in one go.
And I've heard of CISH as well. How does that compare?
Chromogenic ISH, or CISH, uses a standard light microscope, not fluorescence. It's often cheaper, easier for smaller labs to get into. But its big challenge has always been that it's mostly limited to a single color, though dual color is slowly getting better.
For both of these methods, though, there seems to be this hidden enemy that can just invalidate the entire test before it even starts.
Ah, yes, the bane of every lab technician's existence. Pre-analytical variability.
Tell me about it.
I mean, the technical success of a test can hinge almost entirely on steps that happened long before the slide even gets near a machine. Tissue fixation is the #1 culprit.
How so?
Well, if a tissue sample is underfixed in formalin, or if it later gets fixed in alcohol, which doesn't cross-link proteins the same way, you can lose or alter the very things you're trying to detect. An assay designed for formalin-fixed tissue will just fail, or worse, give you a bad result.
Wow, even the temperature matters.
Right, absolutely. Baking the slide has to be controlled. You go above 60 degrees Celsius and you can just destroy many of those crucial epitopes.
So you need some very sophisticated quality control to manage all this.
You do. The gold standard is a built-in internal tissue control. This means staining for, say, a housekeeping protein on the very same slide. If that protein stains correctly, you know both the pre-analytical and analytical phases worked for that specific sample. It validates everything at once.
Okay, so once the tools are defined and you have a handle on the variables, you hit the next big hurdle, the regulatory gauntlet, this parallel path for co-development.
It's a regulatory must. There's no way around it now. And this coordinated effort has to start very, very early. Back in the drug's preclinical phase, you're already doing feasibility studies for the diagnostic.
So by the time the drug hits phase 2 trials, the diagnostic has to be ready to go.
It has to catch up fast. That's when a prototype assay gets rolled out, always labeled investigational use only. And the whole goal during phase 2 is to perform the analytical validation.
Proving that the test itself actually works in a lab.
Exactly. You're proving its specificity, its sensitivity, its robustness, its reproducibility, all the technical stuff.
And then phase 3 is where you prove it matters to patients.
That's clinical validation. You use all the data from that huge phase 3 drug trial to establish the critical cutoff value, the line between positive and negative, and confirm its predictive power. Then, if all goes well, you get simultaneous approval for both the drug and the test.
Let's dig into those validation parameters. What are the key metrics you're looking for, especially for those really complex multivariate assays?
You're focused on 3 big things. First, accuracy. How close is the test result to the real clinical outcome? For a predictive test, that might be how well it predicts metastasis-free survival.
Makes sense.
Second, precision or reproducibility. This one is huge. Can the test give you the same result over and over again within the same lab run, between different runs, and most importantly, between different labs?
And that links right back to what you were saying about robustness.
It does. Robustness and stability. The test has to be reliable even with all those little variations in sample handling, different batches of reagents, and just the passage of time. If it's not robust against those pre-analytical challenges, it's a no-go.
The numbers you see on the efficiency of this model are just mind-blowing. It's not just better science, it's better ethics.
The comparison is staggering. If you use what's called an enrichment design, where you only enroll patients who test positive for the biomarker like they did with trastuzumab, the numbers just plummet.
In a good way?
In a great way. Calculations showed that without the HER2 test, the pivotal trial for trastuzumab would have needed over 23,000 patients. By enriching the population, they only needed 469. Think of the savings in cost, time, and patient exposure to a drug that wouldn't have worked for them.
That's incredible. Okay, let's shift to part 3 and talk about some of these advanced clinical applications, moving beyond just single targets to these multi-gene signatures.
Right. Now we're talking about multi-gene expression profiling, looking at RNA with microarrays, and these tests are so important for figuring out when to pull back on aggressive treatment.
like MammaPrint.
MammaPrint is a perfect example. It's a 70-gene signature, and its main job is to take early-stage, node-negative breast cancer patients and separate them into low and high risk of distant metastases.
So it's about identifying who doesn't need chemo.
Precisely. It identifies the low-risk patients who would get very little benefit from chemotherapy, but all of the toxicity. It prevents massive overtreatment.
And then there's Oncotype DX, which is similar but gives you that recurrence score.
Yes, Oncotype DX is a 21-gene assay that gives you a quantitative recurrence score, or RS. It's used a lot in ER-positive, node-negative breast cancers. But the genius of the RS is that it's not just prognostic.
It's predictive.
It's predictive. Studies have shown that a higher RS score predicts a greater benefit from adding chemo to tamoxifen. So it's not just telling you the risk, it's guiding an active treatment decision.
Let's switch to DNA biomarkers that really get at the drug's mechanism of action. The classic example has to be KRAS mutations in colorectal cancer.
Oh, KRAS is a non-negotiable checkpoint for treatment planning. If the KRAS gene is mutated, it predicts a complete lack of response to EGFR-targeted antibodies.
So drugs like cetuximab and panitumumab.
Exactly. The reason is that the EGFR pathway is like a cascade of signals. The antibody blocks the receptor at the top of the cascade, the front door, but KRAS is a switch further downstream.
And if it's mutated?
It's permanently stuck in the on position. So the antibody can block the front door all at once, but the lights are already hardwired to be on inside the room. The signal bypasses the blockade entirely.
Which is why the drug labels are so strict now.
That's right. In Europe especially, the use of panitumumab is flat out restricted to patients with wild type or non-mutated KRAS.
We also see genomic markers that guide choices around chemotherapy, like TOP2A.
TOP2A, or topoisomerase 2-alpha, is a predictive marker for a class of chemotherapy called anthracyclines. If that gene is amplified or deleted, it tells you how sensitive the tumor will be. And interestingly, about 90% of the time, you find TOP2A amplifications in tumors that are also HER2 amplified.
So they're often linked.
They're often linked, and the FDA has approved a specific FIH assay to measure this, to make sure patients only get that specific toxic agent if their tumor is actually likely to respond to it.
Okay, finally, let's touch on pharmacogenomics.
Yeah.
This is where we shift focus from the tumor's DNA into the patient's own germline DNA and how it affects drug metabolism.
This is all about safety and dosing, minimizing toxicity. A great example is the drug irinotecan, used in colon cancer. Some patients have a polymorphism, a genetic variant, called UGT1A128.
And that affects how they process the drug.
It dramatically reduces the activity of an enzyme that clears the toxic byproduct of irinotecan. So if your activity is low, these toxins just build up and up, leading to a huge risk of severe, life-threatening side effects, especially grade 4 neutropenia.
So what does the test tell the doctor to do?
It's a clear directive. If the patient tests positive, you have to cut the starting dose of irinotecan way down, or maybe even choose a different drug altogether. It's about preventing a catastrophic reaction before it happens.
And we see the same principle with tamoxifen metabolism, but in that case, it's about efficacy, not just toxicity.
Absolutely. Tamoxifen is a prodrug. It has to be activated by an enzyme in the liver, CYP2D6, to work. But some people have genetic variants, like CYP2D64, that make them poor metabolizers.
So they can't switch the drug on effectively.
They can't. They can't convert tamoxifen to its active form, which means they might have worse clinical outcomes because the drug just isn't working at the concentration it needs to. And that information helps you decide whether you need to switch to a different class of drugs, like an aromatase inhibitor.
This whole deep dive really drives home one point. Molecular diagnostics are now the foundation of individualized treatment. We've moved from this one-size-fits-all, often harmful approach to a stratified, molecularly driven oncology.
That's it. Pharmacodiagnostic testing gives physicians the actionable data they need. It cuts down on the twin evils of overtreatment and treatment failure. And that, in turn, reduces patient suffering and, just astronomical healthcare costs.
The path forward seems pretty clear.
The trajectory is set. I would predict that within the next decade, very few systemic cancer drugs, if any, will be prescribed without an associated mandatory companion diagnostic test to guide their use. This isn't science fiction anymore, it's just becoming standard medical practice.
And for everyone listening and mastering this material, we want to leave you with a practical exercise that builds on that co-development phase we discussed.
Here it is. A new drug that targets a protein kinase is just entering phase 2 trials. Its companion diagnostic is ready for analytical validation. Your task is to outline the three key validation parameters: accuracy, precision, and robustness that this prototype assay has to establish. And for an extra challenge, provide one specific real-world example of a pre-analytical problem, like the fixation issues we talked about, that must be overcome to prove that the assay is truly robust.
Until next time on The Deep Dive.